Welcome to Nikolai's Genetics Lessons, and today's question I got from one of my subscribers. Why does well 9, which is here as you see, show more DNA cuts than well 8, which is next to it, after cutting with EcoRV? And here's a question. We conducted a lab where DNA was cut with the restriction enzyme EcoRV. All samples were treated under the same condition, same EcoRV enzyme buffer at 37 degrees of Celsius for 2 hours. However, well 8 shows fewer cuts but larger bands, and the well 9 show more DNA bands than the rest. What could be the reason for this? What could explain why well 9 shows more cuts than well 8 if both samples were processed in the same way? The most probable and common explanation is that the well 8 contained an inhibitor that significantly reduced EcoRV activity, leading to incomplete digestion, fewer cuts, larger bands, while well 9 was free of such inhibitors, allowing this enzyme to cut efficiently and completely, which means more cuts, smaller and more numerous bands. Next, we also can say that probably the reason is that in well 8 sample is less pure, which means it contains also some proteins which also can inhibit process of cutting DNA by enzyme, and may also have different salts which also may serve as inhibitors. We also may not exclude probability that here actually two different species. Sometimes they can be different only by 1 or 2 nucleotides, but those nucleotides may happen at the recognition sites, giving sample number 9 more recognition sites than in sample number 8. So in this case we are going to have more cuts. So more recognition sites may be introduced in sample 9. Another reason for such results can be human error. Actually, for example, in sample 8, a smaller amount of enzyme can be used. So in this case, we also may see fewer bands. So human mistakes also can be a factor. One such human mistakes also can be that DNA in well 8 was not properly resuspended or was clumped, making it less accessible to the enzyme. Another reason for well number 9 to have more bands can be partial degradation of DNA pre-digestion. If the DNA in the well 9 was already partially degraded or contained nicks before EcoRV digestion, the enzyme might cut at its recognition sites, but the pre-existing breaks would lead to an even wider array of fragment sizes, appearing as more bands. However, this would typically manifest as smear rather than sharp distinct bands. Hopefully my explanation will help many of you to understand your gels better, and this is all for today. Subscribe and see you in the next video. Goodbye.